それでは最初のライトニングトークは Google リサーチのリサーチサイエンティストであるビレン・ジェインより Connectomics Mapping the Brain with AI についてお話しさせていただきます。それではよろしくお願いいたします。Mr. Jane, please.
Good afternoon. My name is Viren Jain. I lead the Connectomics at Google team. Achieving a scientific understanding of the brain has proven difficult. Decades of serious investment has produced real progress in some areas, but the reality is we still lack answers to some very simple questions. For example, how are memories stored and retrieved? How do we recognize a face? Why do we need so much sleep? And what goes wrong in Alzheimer's, schizophrenia, and other brain diseases? One reason we don't have the answers to these questions is that we don't have all of the data about the brain that we'd really like. There are 20 times more cells in your brain than there are people on Earth, and then it's the network of the connections formed by these 160 billion cells that enables you to process information from your senses, remember and plan, feel emotions, to be a human. By creating a map of the wiring diagram of these connections, which is called a connectome, we have the ability to unlock new understanding about how our brains work and why sometimes they do not. Mapping the connectome is one of the grand challenges in science, and one that Google Research is uniquely well suited to help tackle. Google has a long history of mapping things. Search was all about mapping the web, and Maps was all about mapping the Earth. Now, we're using our suite of advances in computer science and software to make a map of the brain. We're very lucky to have an amazing set of collaborators from other institutions that share this mission. By working together, we have created new open access tools and data sets that have transformed how neuroscientists do their research. One of the main connectomics efforts we've pursued at Google has been a map of the fruit fly brain. The fruit fly is a tiny organism, but important for biology research. 6 Nobel Prizes have been awarded for breakthroughs achieved by studying the fruit fly. Our collaborators at the Howard Hughes Medical Institute collected electron microscopy images of the fly brain, which we then developed techniques to reconstruct in 3D. The impact has been immense. Hundreds of papers have used this fly connectome to achieve advances in circuit neuroscience and describe how a fly can see, how it can navigate an environment, and how they can learn to associate particular experiences with either good or bad associations. These advances in connectomics have changed the field significantly, such that neuroscientist Larry Abbott now talks about neuroscience as BC versus AC, before connectome versus after connectome. But we don't just work on flies. In 2021, we released the connectome from a tiny piece of human brain, about 1/3 the size of a grain of rice. This video shows the raw data of this human brain sample, starting from a single synaptic connection and then zooming out by a factor of 1,000 until you see a view encompassing millimeters. Even though this was a small piece of brain, it generated over 2,000 terabytes of raw imaging data. The tissue was obtained from a 43-year-old woman as a byproduct of medically necessary surgery who agreed to donate part of her brain to scientific research. After imaging all of this tissue, our AI reconstructed the neurons in synapses using custom algorithms we developed, and then we analyzed the tissue for interesting features. Among the tens of thousands of neurons we reconstructed from the data and millions of synaptic connections, we also came across new structures which we had never seen before. Not just new cell types, but entirely new structures for which we don't really know what their function is. For example, we found that some of the wires will wrap themselves into giant knots. They'll loop around themselves and make a big bundle knot, and we have no idea why this is. Nobody's ever seen this before. And in some sense, that's the beauty of doing science like this. We don't need to be super biased about what to look for. The goal is to provide a data set and resource in which it's all there and people can find and examine different things on their own. We made all of this data public and easy to navigate and encourage you to explore it as well. Use the QR code to check it out or just Google browsable petascale reconstruction. Last year, we launched an important new project. With collaborators and funding from the US National Institutes of Health, we are beginning work towards the first complete mammalian connectome of a mouse brain. The arc of progress here is a little bit like genomics, where the field started by tackling smaller challenges of flies and worms, and eventually reached the human genome. Connectomics is in a much earlier phase of development, shown here by the dashed lines. Mapping a mouse brain, which has 1,000 times more neurons than a fly brain, will drive us to improve and scale our technologies even further, and will teach us important things about how larger mammalian brains work, like our own. The specific part of the mouse brain which we're studying is called the hippocampal formation, and this will help us answer questions like, how does spatial memory work, how do we know where we are, and how does the brain record places and relate them to other memories? The mouse brain connectome could be the largest data set ever collected in biology. This animation compares the data storage of a single Google Pixel phone to the amount of data required for various Connectomics projects. And beyond just acquiring and storing the data, accurately processing all of it is a huge challenge. But that has been Google's unique contribution to the field, developing computational tools that push the state of the art in accuracy and then applying them at larger scale. So what do these tools really look like? One of the primary innovations we've contributed is an approach to automated neuron tracing based on autoregressive 3D convolutional neural networks, called flood-filling networks. We first introduced this in a Nature Methods paper published in 2018. We've continued to refine this technique and shown that it results in a 1,000-fold reduction in the amount of human effort required to analyze the data. Another innovation has been a self-supervised learning technique for Connectomics that learns representations of the data that can be used to identify different parts of neurons, what particular type of neuron is, based purely on patterns in the data itself with very minimal human labeling. This technique is called segmentation-guided contrastive learning, and what last year was published in Nature Methods as well. The method of machine learning employed here is in some ways similar to what is used to train large language models, but more specialized for the type of data we see in Connectomics. More recently, we've worked with our partners to introduce a new method for acquiring the raw data necessary for Connectomics. Last month, with collaborators at the Institute for Science and Technology Austria, we published in Nature a method called LICON. LICON is the first Connectomics method that uses standard light microscopes rather than expensive and complex electron microscopes that have traditionally been required for brain mapping. LICON introduces transformative new capabilities for Connectomics, such as imaging specific molecules of interest alongside synaptic connections, as well as improvements in the cost and accessibility of the research. Finally, we're working with our collaborators at the Howard Hughes Medical Institute in Harvard to bring together multiple modalities of data, combining, for instance, connectomes with real-time recordings of the neuronal firing in larval zebrafish as they respond to their environment. This highly challenging research will enable us to understand how the connectivity of the brain drives its actual function and behavior, and allows us to pursue highly ambitious questions such as whether machine learning could help predict the activity of a neuron or a neural circuit. This year, in ICLR 2025, we published the first results from this project called ZapBench. And we showed how video-based forecasting methods can so far provide the best results in whole brain cellular resolution neural activity prediction, as shown here. So thanks for letting me share my research with you, and just to leave you with one thought, AI is not just about making computers smarter, but also making serious contributions to the most fundamental questions about who we are and what we are. Thank you.
それでは続きまして、Google リサーチのゲノミクスプロダクトリードのアンドリュー・キャロルよりゲノミクストランスフォーミングゲノムサイエンシズについてお話しさせていただきます。Mr. Carroll, please.
Thank you. It's an honor to be here. My name is Andrew Carroll. I'm a scientist, and I've worked for the last 20 years in genomics. It's my honor to be able to tell you about 4 different projects that we've conducted. One is the first creation of the fully complete human reference genome. The second is the ability to diagnose disease-causing variants in record time. The third is the creation of more representative reference genomes for all populations across the Earth. And the fourth is moving into the conservation space, being able to sequence the species of the planet. But first, I want to give some background on what genomics is. So the human genome is a set of 3 billion long characters, and in some sense, this is the original code. You know, we talk about computer code. This is essentially the information that cells and organisms use to do the things they need to do, to make the proteins that drive life and which correspond to traits that you have. So amongst two humans, only about 1 in every 1,000 characters is different. We call these variants, and these are the things which distinguish us from each other. One of the key attributes of genes is that they encode physical structures called proteins. One of the ones I'm showing here is hemoglobin. The job of hemoglobin is to carry oxygen in red cells, and there is a single variant in hemoglobin which can cause that structure to malform in certain conditions, and that can give rise to the disease sickle cell anemia, which is a serious health consequence to people. So this is just one example of the many types of contributions our genetics makes to health outcomes. In aggregate, genetics is responsible for about 30% of health outcomes, and in 6% of individuals, there is a genetic variant which causes some form of serious genetic disease, which you would usually like to act on. So first, how do we read a genome? So it'd be nice if we could just read the sequence end to end, but that's unfortunately not the way things work. So when we get the DNA, it's present in these fragments, and it's our job to identify these individual molecules as accurately as possible, and then to assemble them back together, almost like putting puzzle pieces together on a puzzle box. So one of the first sets of technologies that we built has to do with how do we accurately read the sequences to enable you to construct a new genome. And there was the confluence of these two technologies together. So the first was this technology called circular consensus sequencing, which is a new way of reading longer parts of the genome. And the way it works is there's a single molecule, and what we've essentially done is we've stolen the replication machinery for DNA, and we make copies of this individual molecule of DNA. But because this is a very, very, very tiny structure, the error rate is 12%, meaning it's only 88% accurate. And so what we need to do is have computational methods which correct those errors. And around the same time of this technology, there was an informatics method called the transformer, which is essentially a way of being able to take language information, blanking out spots, figuring out what's important, and constructing kind of the language output. This is the foundation for large language models. We realized that we could repurpose that same strategy to instead blank out the information from a sequencer and train models to reconstruct the accurate results. So this was the first thing that we did was make deep consensus. And like all the software that I'm telling you about, it's all open source, and it's generally available to the scientific community and is applied widely. In the case of deep consensus, we partnered with the company Pacific Biosciences, PacBio, to put it directly onto their sequencing instrument. So that every time the machine runs, deep consensus is what generates the output from that instrument. And because this instrument, since it sequences these single molecules and they're much larger, longer pieces, it enabled us to much better assemble the final human reference, the first complete human reference. So because prior instruments couldn't make sequences that were so long, we were able to, with these larger pieces, then assemble the last 8% of the genome, which had been previously incomplete. And as Varane had mentioned, with the chart of genomics from the effort in 1991, which kicked off the Human Genome Project in 2022, with this technology, the human genome was finally completed end to end in an individual. And this was published in Science. Okay, so once we have a reference genome, what can we do with it? Another technology that we built is called deep variant, which conducts this process of identifying those positions which differ between individuals. So deep variant looks at genomic information in a very similar way to the way a human expert might, but it can do it at a vast scale. Instead of looking at one position or two, it can look at all 5 million positions that differ in the human genome. So deep variant has won multiple awards for highest accuracy in competitions that were hosted by the US FDA. And because AI can learn directly from data, one of the advantages is that when a new instrument comes out, instead of having to write all of the programs to analyze that data, you can train it on examples. So I'm showing you all of the new companies that have put out their sequencing instruments, and in all cases, they put out benchmarks or given best practices in deep variant because it's so rapid to be able to retrain to their technology. Okay, so what can you do with deep variant? There's many such examples. Actually, deep variant has been applied by our collaborators to all of the whole genome sequencing in Biobank Japan. For a clinical demonstration, we partnered with Stanford University on an instrument that could read the genome much faster than previous technology. And one application for this is in newborns. It can often be the case that there is a genetic condition which causes a serious disease, and it's a race against time to find the cause for that disease and be able to give a treatment. So working in a pilot with Stanford, we applied deep variant. We scaled it up onto Google Cloud so it could run very, very quickly. And in less than 8 hours, we're able to go from an individual coming in, having their blood taken, being sequenced. And you can see because of the scaling, almost as soon as variant calling, it starts just 10 minutes later, the variant calls were in the hands of experts for curation and diagnoses were given. In one example, there was a little girl with epilepsy, and she was enrolled in the program, sequenced, diagnosed, and there was a personalized treatment. She was treated, and she was discharged from the hospital in the same day. And usually these processes take several weeks to occur. So this is an example of being able to use AI to very, very quickly identify something that's relevant to a patient. The next project is how we're building reference genomes for not just one individual, but for ancestries for everyone on the planet. So just as you can apply that logic to build one reference genome, we've taken that, and so in the past, the human reference was built from only one individual. And historically, there's been vast overrepresentation of European ancestry in the individuals that are conducted in studies. And on the right, there's this newspaper ad. This is how they found the first people to participate in the Human Genome Project, which was just run in Buffalo, New York, and probably a random town in the United States. People who read the newspaper is not the best way to find representative people of the human population. So working with the NIH, we have applied every aspect of the technology that we've constructed. So we've gone and instead of taking this mix of people that construct a linear reference, we've created complete reference genomes from multiple individuals, and that uses deep consensus for the sequence. We have adapted deep variant to be able to take in as input, instead of just one linear sequence, a graph representation that has the sequences of multiple ancestries. So we can find the ancestry that's the best match for that individual and then be able to give diagnoses. We can show that we can reduce the number of errors by 50% by using ancestry-matched reference genome from the pangenome. And this was published alongside the Human Pangenome Project in Nature. And for this, we also developed a new method, Deep Polisher, which is specifically designed to make better pangenomes in collaboration with the NIH. And for the last story, the amazing thing about genomics is that these instructions power human cells, human sequences, human individuals, but it's really the same sort of instructions that powers everything across life. And we actually have one origin of life, and then subsequently, everything has evolved from it. So we all use the same language. So all the methods that we have developed to analyze and understand a human sample, we can apply to species around the world. So in one project, we're working in Australia to sequence the genome of kelp, which has been devastated by global warming. And what we're trying to do is identify those individuals that have been able to survive the rising temperature in the ocean to hopefully guide repopulation efforts. And in another effort, we've partnered with the Earth Biogenome Project to sequence the genomes of many different species. Hopefully, eventually, we'll be able to sequence every species on the planet. And we've done several of them. We'll shortly be releasing the genomes for all these individuals. There's a number of other genomes that we've done. These are the most kawaii that we had amongst them. And finally, thank you all for your time, and I'll be happy to take any questions that you have.
それでは最後のライトニングトークは Google ヘルスジャパンのクリニカルリードを務めるジョー・レドサムよりヘルス AI についてお話しさせていただきます。Mr. レドサム、please。
皆さんありがとうございます。はじめまして。私は内科医と研究者と Google ヘルスのジョーと申します。私の日本語はあまり上手じゃないだから今から英語を話しましょう。すみません。ありがとうございます。So, as a brief introduction, Google Health is a company-wide initiative, and we have one mission, which is to use Google's tools and technologies to help everyone everywhere live a longer, healthier life. And of course, that spans across all of our products, but especially across the research work. And in particular, we're using AI to sort of support health in many different ways, from accelerating scientific discovery in health and life sciences, to improving the accuracy of things like diagnostics and treatment, to developing tools that people can use to themselves better improve kind of personal health and improve their own health. I'm going to talk about a wide range of topics from multiple different teams today, but I like to start with this, which is a proposal from a team at MIT from 40 years ago, where they outlined a vision for clinical decision support with artificial intelligence. And this was many years ahead of its time, and it wasn't until a few years ago that this work became a reality from a team at Google that released the model MedPalm, which was the first model to pass the American medical licensing test. And this is something that has now been accomplished not just in English or in America, but in multiple countries around the world, including in Japan. And one of the innovations of this paper was not only to look at models in an exam context, but to introduce a framework or a way of thinking about how we evaluate models from open answers, to sort of looking at whether the model answer was in line with scientific consensus, whether it was showed evidence of good reasoning or potential harm or bias. And by putting all of these things together and having experts review them, we were able to come up with a much better understanding of both how well the models were performing, but also where we needed to improve. And you can see even from the paper a couple of years ago, the models were performing on par with clinical experts. Of course, this improvement carried on going, and we've seen the models go from just passing the test to passing at the level of the top test takers. And we can see a few examples in Japanese from these models, where not only did they demonstrate a good understanding of Japanese healthcare, but when there are differences between how medical practice should be in the US versus Japan, we can see that the models, again, even from examples from a few years ago, are culturally relevant and appropriate to Japan. Of course, healthcare is more than just text, and we have carried on improving these models. So a paper from last year, MedGemini, took a wide range of different tasks, so not just text, but medical imaging, genetic information, and really demonstrated that models could accomplish a wide range of different tasks in healthcare. And that allowed exchanges like this shown on the left, where we have a clinician engaging with a model, discussing a particular case, and is able to provide additional information, so in this case an image, to help get a better answer and a better response from the model. And we've carried on innovating in different ways as well. So here is an example where we have a model based on Gemini that is inputting wearable data. And so the user can ask a question about their own health, in this case, how to feel more awake. And the model is thinking about what the appropriate answer should be or what they need to do, thinking about what information it needs, and then writing the code to query the wearable data, coming up with an answer, and then using that answer in its response to the patient to provide a personalized, more appropriate answer to that individual patient. And recently we've seen models getting more and more sophisticated. So this is an example of the AMI model, which it's an example of a model that can interact more directly with a patient in a similar way to how a physician would. And in this case, a patient's asking about chest pain. And in a research setting, the model is replying and asking the appropriate questions, the similar type of questions that we would ask a patient ourselves if a patient came to see us. And ultimately using that exchange to come up with a differential diagnosis. And we can see on the right side of the screen that, again, using this framework of evaluating the model across a range of different areas, the model in green is either as good as, or in many cases, outperforming the physicians in orange. And what's particularly interesting here is in the bottom right-hand corner, we can see that the model's considerably outperforming in terms of empathy. And this is expected. We're telling the model to engage in a specific way, and it's listening to us and following those instructions. But it's really interesting to see that when patients are evaluating the model, that they feel that they respond to that instructions and they respond to the way that the model is engaging with them. Of course, we didn't stop there. So subsequent work gave the model the ability to reason over multiple visits. So here we've got AMI considering not just the diagnosis, but a management plan. It's remembering the patient, it's following them up over time, and it's refining the plan based on how the patient responded. And we can see this, so take a look at what's happening under the hood. And you can see now the shift from a single agent to multiple models working together. So here we have one agent that's communicating with the patient, that's engaging them, that's asking them questions, and then it's passing that across to a management agent, which is then looking up guidelines and querying kind of information in the medical literature in order to better formulate a management plan for that patient. And this is refined and provided back to the dialogue model, which then goes through this process again, and over time, both within the conversation and then over follow-ups as well, we can see this get more and more sophisticated and hopefully a better and better management plan. And you can see from this diagram how this could be expanded to include local guidelines or information relevant to a very particular context, or even to plug in information or models that know and reason more about biology and physiology. Now, health is not just limited to clinics. So we're also exploring life sciences, building on previous successes with tools like AlphaFold. So TX Gemma, shown here, is a new open model that we released. It's based on our TX LLM model that was released last year, and it's tuned on data sets relevant to life sciences. So the model's able to perform tasks like predicting interactions between drugs and targets, or CRISPR repair, or even predicting the outcomes of clinical trials. And this is an open model, so you can take it and you can train it yourselves for your own tasks. But even without that, you can see that the model across many of these tasks is in line or sometimes exceeding state of the art. What I think is particularly exciting about this is not just that model on its own, but how it can fit into a broader agentic framework. So in this case, we have a question which is about comparing two drug candidates, and to decide which of these would be more appropriate for further development in a drug discovery pathway. The core agent, so Gemini in this case, is taking that question, it's thinking about the information it needs in order to answer that, and then it's passing that to TX Gemma, which is coming up with information on toxicity that's relevant to that answer. And then this is used and incorporated into the response back to the user. And this is impressive in a single iteration, but what's really exciting is to think about how this can be, how this works over multiple iterations to compare many, many different candidates together, and ultimately provide a really powerful tool for drug discovery. And we can expand on this even further. So this concept of models working together really came to the fore with the co-scientist model, which is an example, I think, of the limits of agentic AI at the moment, where there are multiple models working together to solve scientific problems. So in this case, a scientist will input a research proposal, co-scientists will come up with a series of hypotheses. There'll be agents that then check that against data sources, against the research literature, considering the likelihood of success. And within the series of agents, they're competing to propose the best idea and ultimately coming up with a ranked list of the most likely hypotheses that answer the information that was inputted. And in several of the test cases here, the model was able to provide create new knowledge rather than just output existing information. And so a couple of examples would be a target discovery for liver fibrosis, where the model came up with potential targets that turned out to be reasonable and potentially new targets for a drug discovery process. And in another one, the model came up with a novel mechanism for antimicrobial resistance, which was tested against a research team that had taken as much as a decade to come up with the same answer. So this is a really exciting area of new research as well. And I like to end here with an example of the Princess Maxima Hospital in the Netherlands. So they're using Gemini, so just this sort of more standard Gemini, rather than many of the models that I've spoke about today, but they're using Gemini to assist with tumor board meetings. Now, this is where physicians get together and consider treatments for people with complex cancers. And here, Gemini is providing literature evidence. It's searching through information to help the physicians better plan and better use that information in the treatments that they end up giving the patient. But I think this is really exciting, one, because it's a great example of how humans and AI can work together for the benefit of patients, but also to imagine how this will look when it incorporates many of the models that I've spoke about today, where the models are not just looking up current literature, but we have a series of agents that understand healthcare and medicine, but also biology and also physiology and pharmacology. And they're working together, not just pull up additional existing information, but to really reason about personalized treatments for patients. And of course, this is still a future vision, but it's one that we're really excited to work on with everyone together. So thank you very much.
それでは続きましてダイアローグセッションに参ります。テーマは健康的なエイジングのためのウェアラブルデバイスと AI 活用です。本日は東京大学大学院新領域創生科学研究科スマートヘルススクールスクール長久常達弘様をお迎えし、Google AI リサーチ&コアパートナーシップ日本リード加山弘樹と対談をさせていただきます。それでははじめに加山さんお願いいたします。
皆さんこんにちは。 Google の加山と申します。長いセッションお疲れ様です。ここからはですね、ダイアログセッションですので、皆さんと会話をしながら進めたいというふうに思っております。よろしくお願いいたします。それではここまでですね、本日は AI 技術の発展が人類生物学というところの研究開発にどのような影響を与えてきたか、どのような貢献をして革新的な効果を達成しようとしているかというところを伝えさせていただきました。でここからはですね、舞台を世界から日本に移させていただきまして、 Google が東京大学と行った高齢者の健康改善におけるウェアラブルデバイスの有効性の研究とその結果についてお話しできたらと思います。で本日のイベントの冒頭において、弊社の日本法人代表の奥山から発表させていただきました通り、この研究によりウェアラブル端末が高齢者の認知機能の向上や心身の衰えであるフレイルというところにつながるということが明らかになってきました。そのテクノロジーが健康的なエイジングを力強く後押しする可能性を示唆したと、そういう研究ですけれども、その研究プロジェクトをリードしてくださった東京大学の久常先生と、この研究に至った経緯だったり課題、そして研究がもたらす社会的意義というところに焦点を当てて深掘りさせていただけたらというふうに思います。それでは久常先生、ステージによろしくお願いいたします。皆さん拍手でお迎えください。
紹介いただきました東京大学の久常でございます。
ろしくお願いします。先生と研究をいろいろ通させていただいて、たくさん大変なことがありましたけれども、やっと結果が出てですね皆さんにこうやってお伝えできる瞬間がやってまいりました。でまずはですね久常先生の方からぜひ研究の目的だったりとか、あとは調査の方法ですね、でどのような結果が出たかというところを簡単にご説明いただけたらと思いますが、お願いしていきますでしょうか。
スライド。
はい。
私たちはここにあります三井不動産の明日っていうところと長く共同研究をしておりまして、そこには高齢者の方の会員の方がたくさんいらっしゃると。それでジョーさんとこのウェアラブル端末を使った研究を始めるにあたって、最初はですねやはり欧米肥満っていうのがやっぱり健康上のトラブルだろうっていうところで、そこに焦点を当てた研究をしようということで、数ヶ月準備をしたんですけど、よくよく考えてみますとここにありますようにご高齢の方、太ってる方は誰もいないと日本の場合はやはり痩せてる方がもう健康的には問題で、ですからもう根本からこの研究のデザインを変えて痩せてる人を救う研究をしたいということで、次々出てきますタンパク質を補給するっていうことも含めたデジタルデバイスを使った高齢者の方の健康改善に対する研究を行いたいということを目的として実施いたしました。じゃあ次に、はい。ですので、まずはこの栄養っていうところ、タンパク質これ15から 20% を取ってほしいっていうことであったり、運動としては1日あたり8000歩、睡眠時間はいろいろあるんですけど、だいたい6時間から9時間ぐらいがいいのではないかということで、これはパーソナライズされたお知らせを先ほどの AI を使って皆さんに定期的にお伝えすると、それで健康的なところが改善できるかということに関して認知機能を測ったり、あるいは身体の機能を測ったり、メンタル的なところをお聞きしたりということの研究を行いました。ここにありますように研究参加者の方が300人いらっしゃって、それはなかなか大変な研究で、この先ほどの説明会50人ずつの方を6回集めて、ジョーさん、加山さんにも柏まで来ていただいて、ご高齢の方にまずスマホの使い方みたいなところから始めてご指導を申し上げたっていうところで、それでは研究はですね、今日はもう本当触りの部分ということになりますけれども、まずそのフレイル的な方を集めたので、そこに対してはまず効果がありました。しかし驚いたことにこの認知機能っていうものがですね、この半年間その栄養、運動、睡眠の介入をしますと、すごい有意差で良くなってるっていうことが分かりまして、これが大きな驚きで、タンパク質の摂取量はお知らせをしたっていうこともございまして、1日あたり7グラムぐらい増えていたと。でもう一つの驚きは、歩いてくださいって言ったんで、そこが伸びるかなと思ったら、このじっとしてる時間ですごい差になっているっていうところで、活動的な、元気なお年寄りになってきたんじゃないかっていうところがございます。ここの結果のところには示してませんけど、MCI 的にちょっと認知機能が落ち気味の方も半分ぐらいいらっしゃいました。その方々にもすごく効いていましたし、ですからこの健康増進っていうのは、単なる健康増進からさらにはその認知症を予防するみたいなところも含めて今後展開できるのではないかと考えております。
ありがとうございます。今もう先生がご説明いただいたところではありますけれども、今回介入群と対象群というところに分けさせていただいて、介入群の方々にはフィットビットをつけていただいたと。で6ヶ月間走らせていただいて、そのフィットビットで測られる内容、運動だったりとか睡眠だったりとか、そこをベースにアドバイスを送らせていただいたという形になります。アドバイスベースで、皆さんの行動変容だったりが引き出されて、このような結果が出てきているのではないかなというところを研究結果として発表させていただいたという形ですね。はい、ありがとうございます。この研究から先生のぜひご意見を伺いたいと思うんですけれども、 どのような、新規性がある効果、社会的意義も含めてどのようなところが、新しいのかというところはいかがでしょうか。
はい、まずは結果のサポートをしていただきましてありがとうございました。その得られたところも先に話してしまいましたけど、そのフレイル的なところに効くっていうことは最初予想した通りだったと。やはりびっくりしたのはこの認知機能のところにインパクトがあったっていうところで、私、この研究に加えてアルツハイマーマウスの研究もしておりまして、それで栄養とか運動とか改善すると、不思議なことに脳の方に効くんですね。それがこの人でもしっかり結果として出てきたんだなっていうところで、ここの部分は強調してお伝えしたいかなと思います。
ありがとうございます。まさに先ほど先生もおっしゃられましたけど、最初やはり運動機能だったりとか、筋肉量だったりとか、そのあたりに影響が大きく出てくるんじゃないかなっていうところを我々予想して研究を始めたんですが、認知機能というところに顕著な変化が出てきたというところが非常に面白い研究だったのかなというふうに思っております。ではですね、次の質問ぜひさせていただけたらと思うんですけれども、これからこうやって広げていくことによってこの日本、今超高齢社会と言われておりますけれども、その超高齢社会にどんな影響をもたらすことができるっていうふうに思われますでしょうか。
やはり、ご高齢の方とお話していると、よく寝れて今日も一日元気だなと思える日はもう素晴らしいと、もうそれだけでいいっていうような方も結構多くいらっしゃって、ですから、このように、ウェアラブルデバイスで、自分の健康を高めるってことは、元気を高めるってことにつながるんじゃないかなと思いまして、それで、シニアの方の元気が高まると、全体も元気になるんじゃないかっていうところもあって、そういうことに期待をして、研究活動を続けていきたいと思っています。
ありがとうございます。まさに生き生きした高齢の方々がいらっしゃると、それこそ経済の効果というところも大きいんじゃないかなというふうに思います。皆さんはやっぱりいろんなところに社会貢献したいとか、それこそ再雇用ですね、もう一度仕事をして何か経済を回したいという方々も増えてくると思いますし、それこそ研究というところにもう少し時間をかけてどっぷりやられるという方もいらっしゃるかもしれません。そういうところで本当に活発なシニア層、生き生きしたシニア層が多くなるというのは非常に有意義じゃないかなと私も感じます。では次の質問させていただきたいと思いますけれども、実際にこの技術、やはり今回研究させていただいててもいろんなハードルがありました。今後広げていくために乗り越えなければいけない課題っていうのはどういうものがあると思いますでしょうか。
そうですね、結構大変だったのはこの食事のところをトレースするっていうところが難しくて、例えばタンパク質の量、本当はこのフィットビットで測れるのが一番いいなと思っていて、今後5年10年のうちに測れるようになったらいいなっていうこともありますし、あとはやはりこういうサービスを届けていただく、いろんな会社の方と連携をして、社会実装に結びつけていきたいということで、後ほどご関心のある方にもう少し説明しますけど、私たちこういう社会人スクールとかもこの秋から始めるっていうことで、皆さんと連携して社会実装のところにも取り組んでいきたいと考えております。
ありがとうございます。まさに先ほどおっしゃった栄養のところは、いろんなソリューション、例えば写真で撮るだったりとかあると思いますけど、写真で撮るソリューションだと、例えばおにぎりの中に梅干しが入っているのか焼肉が入っているのか、それによってカロリーだったりとか栄養素は大きく変わってくると思います。そのあたりも非常に難しい、これから科学技術の発展が必要なところだなというところを本当に同意されるのと、あと先生こういうふうなセッションですねやられているというところで、皆様高齢者の方々に対する研究だったりとか社会実装というのをどういうふうに進めていけばいいかっていうのをご一緒に考えられるといいかなと、そういうコミュニティの活性化っていうのも非常に重要かなと私も思います。ありがとうございます。ではそろそろお時間なんですが、最後にですね、先生こういう共同研究させていただいて、Google に今後こうしてほしいとかこういうことを期待するみたいなことがあればぜひ教えていただきたいんですけれど、いかがでしょうか。
一つはですね、日本初のっていうことでこういうことをやってるわけですけど、ぜひ Google といったら世界規模の企業ですので、そういう、広くコラボレーションっていうのが進めていけたらいいなっていうことと、あとはこの、デジタル的なアラートをやっていったんですけど、やはり毎回少しずつ違う生成 AI を使って、日常会話のようなアラートができるともう少し訴求力があるっていうところで、そこで先ほどジョーさん言われた生成 AI を使ったサポートっていうのを展開していきたいかなと思っています。
ありがとうございます。ぜひそのような時代が来るように頑張らせていただくと思います。
以上をもちまして、ザ・ビヨンドシリーズリサーチアット東京ブレイクアウトセッション AI for Health Science を終了いたします。